Hello, good afternoon. I am Dr. Jose M. Oclarit, a PhD in Applied Biochemistry and Molecular Biology at Hiroshima University in 1994. In 1996, I was also declared as an outstanding young scientist of the Philippines in the field of biochemistry by the National Academy of Science and Technology, Manila, Philippines. Okay, this afternoon, I would like to make this broadcast regarding the PCR testing. PCR stands for polymerase chain reaction. This was discovered by the scientist Dr. Karim Yulis, and PCR is all about magnification of the genetic materials, either RNA or DNA, no matter how small the amount, the genetic materials. Through the use of PCR, it can be magnified million times or several times using the PCR machine. The PCR machine is also known as a thermal cycler. The principle behind this PCR machine is that the DNA, being a double-stranded molecule, will be subjected to thermal cycle. Like the first of all, the DNA should be made into single-stranded DNA, and by melting it at a temperature of about 95 degrees Celsius. After that, it will be cooled off to about 65 degrees Celsius in order for the primer of a particular genetic material to anneal with or to pair with the template DNA or material present in the surrounding, and then it will be elongated using a Taq polymerase or a DNA polymerase that would now synthesize new DNA molecule using this particular heat stable enzyme called Taq polymerase, DNA polymerase. And then with the supply of the free nucleotides like thymine, guanine, cytosine, and adenine, it will now be elongated at a temperature of about 65 degrees Celsius, and then it will be in a buffer, and then it will continue until it will be able to make a new copy of the template DNA or RNA. And then it will be repeated several times. So one thermal changes from 95, 65, 72, then 0 again, and then back again. You call it thermal cycler. In this particular case, this PCR technology is able to magnify or what you call multiply the number of copies of genetic material several times in the order. Usually, in my experience, I am able already to see the DNA material at about 24 cycles of thermal cycling PCR. 24 cycles. So the higher the cycle, the higher the magnification, the more the materials. And then later on, the PCR products or the multiplied DNA products will now be subjected to what you call the electrophoresis in order to see the PCR product. So the electrophoresis will be able to segregate now or identify the magnified DNA material or RNA material. So the advent of this PCR is that the PCR will be able to magnify any genetic material from the surrounding. Thus, using this particular principle, magnifying the DNA outside the cell using this PCR is actually a radical method by which they were able to exploit this as a diagnostic tool. True indeed, PCR can be used as a PCR diagnostic tool. However, the sensitivity and the efficacy of the PCR as a diagnostic tool lies on the specific primer that will be used in order to magnify the genetic material. It is true indeed that you can isolate genetic material from the surrounding using a PCR, using the the primers of a particular specific organism. So, the most crucial point in PCR analysis or PCR testing is in the use of primer. So, in order for you to identify really and see if the genetic material is present in the surrounding, like a swab or on the table, on the nose, on your tongue, on your saliva, and in your blood, or so on and so forth, the success of this PCR as an identifying what you call diagnostic tool relies on the specific primers. So therefore, the reliability of the PCR relies on the specific primer. However, in the case that is being used now, I just wonder why they will be able, they cannot get a specific primer for COVID because COVID keeps on mutating. You cannot make a specific primer for those organisms that keeps on mutating. Otherwise, you will just be using what you call as a universal primer. What do you mean by universal primer? Meaning, these are the sequences of genetic material which are common to all kinds of viruses. So, I suspect that this PCR testing that they are using just only a universal primer for COVID. Meaning, universal primers for coronaviruses. Meaning, even if it is not COVID, for as long as you have a virus, you will come out to be positive for PCR analysis if your primers are not specific. So therefore, the reliability of the PCR machine lies on the specific primer for the specific causative agent. So although I believe in the PCR as diagnostic tool, as I said, I repeat again, the reliability of this PCR test machine lies on the use of the specific primers. Therefore, in order to be successful, to be able to identify the genetic material, you should have a very specific primer for a specific organism that is solely identifying that particular organism. Otherwise, you will have a false positive or false negative. Thank you very much and good afternoon.